All right, our next presenters are with Aldevron. They are Nate Russart, Principal Scientist for R&D, and Tanner Dockendorf, Innovative Product Manager. Welcome, gentlemen. It's all yours.
Wonderful. Good morning. Thanks to Perry and the team here, and thanks to everyone who's joining into the webinar this morning. We're starting off with a quick poll question, so take a look at the screen. The question is, what is your organization's current level of engagement with synthetic DNA technologies for mRNA production? Dive into it here. Like Perry said, my name is Tanner Dockendorf, Director of Innovation Product Management for Aldevron's DNA business. This morning, we're going to be speaking about Aldevron's newest capability we're launching into the market, our Alchemy Cell-free DNA technology. Before we get into that, I want to take a little bit of time here to introduce you to Aldevron as an organization in case you're not familiar. Then we'll hop into the overview of our Alchemy technology. For over 25 years, Aldevron has been committed to supporting our customers and the markets they serve. We strive to be more than a CDMO, rather a partner focused on providing innovative solutions to support our customers' goals and our own company vision, a world where every cure is possible. Since 1998, we've grown and evolved our business to meet the pace and needs of our customers, and we continue to innovate, which is exemplified by the topic we're going to be talking about this morning. For folks who aren't familiar with Aldevron, we are a CDMO offering services built around three key platforms, those of DNA, RNA, and protein. And our manufacturing capabilities here range kind of across the board from standard plasmid DNA production in E. coli cells, linear DNA template production, to IVT enzymes and mRNA drug substance, to more innovative solutions such as our proprietary nanoplasmid technology and novel gene editing enzymes. Importantly, we don't just offer standalone products within each of these verticals, but really the ability to provide modality-based solutions by knitting together service offerings and products to drive value and convenience for our customers. For example, RNP complexing and for gene editing applications and sequence to vial LNP encapsulated mRNA drug product. We've listened to our customers and understand the importance of being able to support a broad range of services within a single modality, so built and expanded these capabilities to do just that. Oops, toggle slides here. Aldevron's footprint is currently split between Fargo, North Dakota, and Madison, Wisconsin. The Fargo site has two campuses, Advance and Breakthrough, which house our corporate headquarters, as well as our R&D and research grade operations for both DNA and RNA, as well as our GMP operations for all platforms. Madison is home to our protein assets largely, specifically our R&D function, process development, as well as our ISO 13485 manufacturing capabilities. Between the three campuses represented here, we have intentionally built out capacity so as not to be the bottleneck for customers and have the capability to expand capacity as needed to meet the demands of our customers. Now, a lot of the services I've referenced thus far have fallen into the bucket of custom manufacturing, which is really Aldevron's bread and butter. But in addition to the custom manufacturing solutions we recognize the need for speed, accessibility, and convenience during drug development. As such, we've developed suites of ready-to-ship catalog solutions spanning all of our platforms, such as gene editing nucleases, wild-type and mutant IVT enzymes, various helper and capsid plasmids for viral vector production, and standard cloning and reporter constructs for basic research needs. All of these products mentioned are available at a research grade level, and many are also available at our intermediate quality grade, GMP source, as well as full GMP, all to enable continuity of supply as you progress from preclinical to clinical and beyond. These products are really designed to accelerate development and to take some of that burden off of you and your team's shoulders. Here's just a quick look at some of our facilities. Not quite the fly-through video we just saw, but a look at some of our GMP clean rooms and our Breakthrough campus in Fargo. All right, then to summarize the company overview portion of the presentation here, at Aldevron, we've really created an ecosystem of capabilities uniquely suited for supporting our customers across service levels, phases of development, modality, and product type. And we've also invested in the supporting capabilities to provide success along the way, thinking from a quality, regulatory, storage, distribution, stability perspective. In addition to our internal capabilities, we're also part of a larger Danaher life sciences and genomic medicines network with the capability to partner across sister operating companies for access to various services, products, and equipment. As part of this network, we're able to provide a robust and broad portfolio of capabilities to customers in the space. Now, I'd like to take the opportunity here to pivot to the main topic of our presentation. I'm very excited to share that Aldevron launched our newest service offering just last week, our Alchemy Cell-free DNA technology. We really look forward to bringing this technology to our customers at the research grade level today, and later I'll touch on how we plan to grow this service in the near term with an eye towards clinical and commercial support. I'll hand it over now to my colleague, Nate Russart, who will take you through the next part of our presentation.
All right, thanks, Tanner. Thanks for teeing us up on a little bit of the background on Aldevron. Thank you to Perry and the team for having us listening. So as I said, my name is Nate Russart. I'm a principal scientist here at Aldevron, and I've been able to do a lot of the work on the development of our cell-free DNA technology, and we're very excited, myself and my team, to bring this out and launch it onto the market. Okay, so why cell-free DNA? Why enzymatically produced DNA? You know, one of the strong benefits of the cell-free technology is the reduced linear DNA manufacturing timelines. If we think about a traditional plasmid DNA workflow, we have transformations, grow-ups, plate streaking, colony picking, sequencing colonies, growing up and producing a supercoiled plasmid, purification, QC, back into a linearization reaction and additional QC research programs. On the other hand, with enzymatically produced DNA, we can do amplification and quickly into a purification, linearization and purification in the QC. So we're able to reduce our manufacturing timelines by up to 50%, allowing you to get your material back faster and continue on with your programs. Okay, a little bit about the process and how it works. So it begins with a template that goes into an isothermic rolling circle amplification reaction to amplify it. That template is going to be a plasmid DNA template. So it's going to be the same starting material that you would normally send us if we were going to do a traditional plasmid DNA manufacturing process. Following amplification through that rolling circle amplification process, it goes through a linearization using the same restriction endonucleases that you would use for traditional plasmid DNA once again. And then you would go into a simple purification, QC, and ready to go into your IVT reaction. You know, other advantages of the cell-free technology, because we're not using those E. coli cells, we're eliminating any of the cell-derived impurities that are associated with them. Things like genomic DNA, residual RNA, host cell protein, and endotoxin are virtually eliminated through this process. Additionally, because there's no selection needed, there's no use of antibiotics, so we don't have to worry about residual antibiotics in our product as well. Okay, so our final product is a linear double-stranded DNA. All right, so there's never any closed ends as part of our process. So this means there's no need to remove those ends that could reduce, that could produce double-stranded RNA. You might have to develop assays to ensure that those closed ends are removed. We never have a closed-ended step. And then it's the exact same sequence as your plasmid. So if you're transitioning from a plasmid DNA workflow to our Alchemy DNA, you don't have to develop new assays, new protocols, or anything like that. It's the exact same sequence. All right, so as you can see, it's well suited to fit right into your mRNA workflow. We developed this process with mRNA production in mind. This was not, developed to produce a DNA product. It was developed to go right into an IVT reaction. And so as we worked through developing this, we did work with industry collaborators to evaluate our material and ensure it was behaving as expected and had high quality as we are accustomed to. So the first thing I want to talk about is the integrity of both the DNA product and the resulting mRNA that's produced from it. And so here we have two constructs, construct 1 on the left, construct 2 on the right. So the orange bars are going to represent a traditional plasmid DNA template, and then the mRNA produced from the plasmid DNA. And then the yellow bars are going to represent the Alchemy DNA template, and then the mRNA produced from that Alchemy DNA. And so here the integrity is measured by capillary gel electrophoresis. And we can see across both of these constructs, there's no difference in the integrity or the full-length product of the DNA template. And in this case, the mRNA produced from the Alchemy DNA, the integrity was a little bit higher. I wouldn't say that's going to always be the case, but you're going to see at least comparable integrity from the mRNA produced from Alchemy DNA. Okay, our Alchemy DNA product allows for IVT process consistency. So once again, we offered, through this development process, we offered our collaborators both traditional plasmid DNA that was produced in our research grade manufacturing system, as well as our Alchemy DNA. We didn't give them any, conditions under which to test it. We said, treat it as if you would pDNA. And let us know how it works. So one of the things that was that came up and a concern was, is this removed through DNase treatment as readily as pDNA is? And so here we can see a collaborator measured through qPCR the remaining levels of residual template after their IVT and DNase treatment. And we see that the cell-free process, the cell-free Alchemy DNA, had lower levels of template in their final product when compared to plasmid DNA. An additional advantage of our Alchemy process is that because we are amplifying that entire plasmid, there's no need to develop a new qPCR assay, which often target those backbone regions of the plasmid for consistency. Okay, and then finally, I want to talk about expression. So this is, probably the most important thing, but the thing that's also kind of like the table stakes. We had to make sure that the mRNA produced through our Alchemy DNA is going to function and perform just as well as mRNA produced from plasmid DNA. So in this example here, a collaborator had a self-amplifying mRNA construct. So it was a very large construct that expressed vaccine antigen. They expressed this into HEK cells and then measured the expression by flow cytometry. And we can see that there's no difference in the expression of that antigen when using traditional plasmid DNA template or Alchemy DNA template. Okay, so as you can see, the Alchemy DNA has all the quality attributes that we would expect when we're using traditional plasmid DNA template. And you get your material back in, half the time. So it's kind of a win-win. And so we're very excited to launch this into the market, and we hope you'll give it a try. And with that, I think I will hand it back over to Tanner.
Thanks, Nate. As I mentioned earlier, this launch is specifically encompassing our research-grade cell-free DNA capability. As we look to the future, we definitely understand the need to scale and grow with customers. Like I mentioned, that's been part of the core philosophy of Aldevron since day one. So we are intentionally investing in this Alchemy platform to do just that. To that end, we're in the process of readying the counterpart GMP offering to Alchemy with the aim of launching our GMP capabilities by the end of 2025, so we can offer truly a full kind of continuum of service offerings from research through GMP, so the customers leveraging this technology can use it in the early stages of clinical and follow that through all the way to a commercial product. We'll leverage our GMP facilities and capabilities and capacity, which are proven to successfully support clinical and commercial products today, and apply these experiences to this new technology. I want to draw your attention to this particular slide. This is kind of a tying it all together, kind of big picture look at what the Alchemy technology can mean for our customers as we think about stitching it together, again, in kind of a modality focus with our mRNA capabilities already at Aldevron. Really thinking about the seamless transition of this cell-free, high purity, quick turnaround Alchemy input into our IVT process, into our LNP encapsulation, into a final mRNA drug product in one contiguous workflow. That's, of course, where we're heading, and as you can see, kind of the future of this type of manufacturing. So I wanted to kind of leave this as a teaser for the future as we think about how we're putting this all together for the market. We're really excited about the potential value here and would love for folks to test this out on their workflow. So if you have questions, please reach out to us. I think we have some time here for questions as well. But thank you for your time and attention, and I will hand it back to Perry.
Thanks, gentlemen. Really fascinating stuff. So I understand that at the JP Morgan Healthcare Conference, mRNA was among the hottest of topics. So now you're introducing Alchemy. Brilliant. This is really good stuff. We feel very special that you're using our platform for that. We have a question from an audience member, pretty specific. What is the sequence error rate of the synthesized DNA? And then what is your typical manufacturing turnaround time?
Yeah, it's a great question. I'll maybe take the second half of that, and then I'll let Nate speak to the error rate, as that's obviously a hot topic in the synthetic DNA space. As far as turnaround times for the research grade offering that we have launched, to Nate's point, the big value here, one of the big values is really speed in comparison to all of the steps we have to go through for E. coli-based linear production. So as we're talking about most scales at research grade, really around a week of time from start to finish, actual wet lab time, thinking about the amplification, the linearization, purification, and QC testing.
Yeah, and then to the sequence error rate, so that's a great question. I think something that comes up a lot, right? And so these enzymatically produced DNAs are not going to have the same sequence fidelity as traditional plasmid DNA has. I mean, there's just not the error correcting mechanisms that are available in the E. coli cell. That being said, the error rate is low enough that you don't see a difference in functionality of this material. So it's very difficult to measure the exact error rate, but somewhere in the range of 10 to the negative 6, 10 to the negative 5, using standard methods is a good ballpark of where the error rate is. We'd love to talk more about that with you if you wanted to connect offline or something.
Okay, thanks, gentlemen. Yeah, you can see that question in the Q&A box, so you can follow up offline. It's very interesting. Overall, so Tanner, you talked about timelines, but overall, using this technology, what kind of timeline reduction in efficiencies are you experiencing?
Yeah, Perry, I think that's a really good question. Obviously, the bulk of the timeline efficiency today is in the template production, the DNA template production. Like we said, over 50% reduction there compared to standard. I think where we have another benefit compared to other solutions in the space, to Nate's point, is we are producing an end product that is directly can plug into the IVT reaction. So omitting any manipulation steps from template to IVT, which adds some efficiency. And then, like I mentioned, in an ideal future state, trying to stitch this all together into one uniform process for that efficiency.
Fascinating. So Nate, did you want to add anything to that?
No, I think you covered it well.
Okay. I don't want to miss anything. Okay, so if somebody has an existing mRNA workflow, can Alchemy be integrated into that and still get the benefits of the things you're talking about?
Yeah, I can cover that. So exactly, and I think that's how we developed it in mind, is that it would be a direct transition from using their traditional pDNA to the Alchemy DNA as a template for their IVT reaction. So there's no difference in the sequence at all. There's no retrofitting into a different backbone. And the fact that the backbone is still there means that there's no difference in the copy number. So it's really just a plug and play. Instead of your traditional pDNA template, you can go with the Alchemy DNA and expect similar results.
And so as somebody's working with you on a project, what kind of general support do you give them? This is kind of unique, and so it may be a non-traditional support. What does that look like from the client's perspective?
Yeah, obviously this is a new offering, so we just launched this in the past week, and so we did a lot of work with our collaborators that worked on us through this development process. And as we start producing this, we will be working alongside people, and we need that feedback as well. But we anticipate that it will work just as well, and that's what it's shown through our development process. So, we are here to work with them if issues come up, but we don't anticipate that.
Okay, and finally, what other kinds of applications do you foresee with this technology?
Yeah, that's a good question, Perry. Obviously, with the kind of focus on linear template today, anything mRNA is fair game. We're also looking at other forms of synthetic DNA as well to serve other applications in the market, but a little premature on that just today. Excellent.
That's great. I feel like we're living in the future. This is a futuristic presentation, but still very real. Thank you, gentlemen. Just a great presentation. Very interesting technology. Alchemy cell-free DNA technology. Fascinating stuff. Thank you very much for being with us, Nate Russart and Tanner Dockendorf from Aldevron.